Thank you, Dr. Ihara, and thank you for organizing committee for giving me this opportunity to talking about the CARASIL. This is my COI. And CARASIL is first reported from Japan. It's a cerebral autosomal recessive idiopathy with subcortical infarct and leukoencephalopathy. The CARASIL has three distinct clinical features. One is a leukoarosis, and also the spondylosis and alopecia. These three characteristic features can distinguish the patient, can diagnose the patient as a CARASIL. The MRI findings very resemble to the CARASIL, but the difference in the advanced stage, sometimes the linear high intensity observed in the midbrain and middle cerebral pedunculus. This is a clinical frequency of the CARASILs. One of the big difference to CARASIL is the frequency of migraine is very low in the patient with the CARASILs. And the frequency of alopecia is not 100%. And therefore, without alopecia, we cannot rule out the possibility of the CARASIL. And the spondylosis gait disturbance is frequently observed in the patient with the CARASIL. And the stroke episode is a little bit lower than the CARASIL. And for the MRI findings, the leukoarosis of the MRI, beginning from the frontal lobe, in advanced stage, it extended to the occipital lobes. And one of the characteristic MRI findings is a high intensity lesion in the external capsule is well observed in the CARASIL patient. But in the cerebrums, the high intensity is not observed in the CARASIL patient. Also, the atrophy is progressed by the progressive. And the pattern of the atrophy is not so specific. But the central type of the atrophy is progressive in the late stage. The gene of the CARASIL is HTRA1, high temperature requirement A1. The function of this gene is the same protease, and it inhibits TGF-beta signaling. And one of the characteristic features of the HTRA1 is it's a trimer formation. For the activation of HTRA1, this trimer formation is take a very important role. The trimer formation is necessary to activate each HTRA1. And after we identify the mutation of the HTRA1, we easily find out that most of the mutation, the protease activity is decreased. And thus we can speculate that maybe the loss of the activity of the HTRA1 called the CARASIL. But one of the mutation, R370X, retains the protease activity. And that's a problem. And therefore, we're thinking about, we focused on why this mutation caused the CARASILs. This mutation fulfills criteria for the degeneration by nonsense-mediated messenger RNA decay. And therefore, we obtained the fibroblasts from the heterozygote and find that the mRNA level is markedly decreased in the fibroblast of mRNA of the heterozygote. And therefore, we conclude in these mutations, mRNA is degraded by NMD. Therefore, the CARASIL is caused by a loss of HTRA function. During the search of the patient with a mutation of the HTRA1, we spontaneously find out about 5% of Japanese severe CSVD case with or without family history has HLP lesion. At the same time, in the European peoples, about 5% family with small cell vessel diseases cause a mutation in the HTRA1. It pointed out maybe some mutation causes cerebral small vessel disease even in the heterozyte. This is a result of the Japanese population. We collected the DNA samples from the Japanese individuals who has a severe leukoarthritis and regardless of family history and 17 years old. Among them, about 25% has a CARASIL mutations, but surprisingly, about 8% individual has a mutation in the HTRA1 with heterozygote. Just about 2% is a homozygote mutation of the HTRA1. Therefore, in Japan, nearly 10% of the individual caused by mutation of the HTRA1. This is a representative MRI findings of the symptomatic carriers in the HTRA mutations. This is the MRI of the CARASIL, and from here to here is the MRI of the heterozygote. As you see, the leukoarthritis is not so marked as CARASIL, but the atrophy is marked compared to the CARASIL. And also, I would like to point out that the temporal tip involvement is not observed in these individuals. Therefore, this is very difficult to distinguish these individuals from the sporadic CSVDs. That's a problem to thinking about the possibility of the mutation of the AT1 in just CSVD patient. Some of the patients also show the alopecia and spondylosis, but the frequency of the alopecia and spondylosis is very low in the heterozyte. And very interestingly, this is a summarize of, from the literature. Until now, we have accumulated about 46 individual symptomatic carriers and 28 CARASIL patients. And in the symptomatic carriers, about 76% is a male. We don't know why the male predominance is observed in the symptomatic carriers, but it's very remarkable. And in the symptomatic carrier, tend to have the risk factor hypertension. About 50% of the individual has hypertension. And also age at onset as relatively elderly, about 54 is average of the onset. Compared to the CARASIL is the 30s. Therefore, the male predominancy and they tend to have the hypertension and more elderly onset. Regarding the clinical features, gait disturbance and is frequently observed, but not so high as CARASIL. And our picture is only 13%. That's we cannot rule out heterozygote mutation without our pictures. The Lumbago is 60%. So this shows a more mild phenotype. But one of the interesting things, these individuals has a more frequently episode of the ischemic stroke. Among the CARASIL patient is 34%, but the symptomatic carriers, they have the 60%. This graph shows the location and the number of the mutations which have been identified in only symptomatic carriers or only in CARASIL. You can easily notice there's a two peak. The mutation in the find only in the symptomatic carriers. This peak is located at this region. This red line region, is very important to form the trimer in the H01. Now, the another peak is located in this red line and this green box. And this red line is also very important to form the trimer. And also this line, this amino acid, is very important to activate the besides the HTL1. Therefore, the mutation which found in symptomatic carriers is tend to locate at the amino acid, which is very important to the mechanism of the trimer-dependent active cascade. So this animation of how the HCl activate each other. When the substrate binding to each HCl1, this HCl1 activated by beside. Therefore, the trimer formation is very important to activate all of the HCl1. And the activation cascade, L3 and LD domain takes the important roles. So the intact LC and LD domain is necessary to activate the neighborhood HCl1. Thus, we investigate which domain has a mutation and the trimer formation availability for each mutant HCl1s. And very interesting, most of the mutants which have been reported in the symptomatic carriers do not form trimer or has a mutation in the LD or L3 domain. It's very clear. But some of the mutation has forms a trimer and also has a mutation at the protease domain. It's not L3 or LD domain. And also we found some mutant which has a frame shift or the nonsense mutant also shows cerebral small vessel disease even in the heterozygote. In these individuals, these RL, the protein obtained from these RL may be degraded by the nonsense mediated messenger RTK, and therefore at least 50% of the protein activity they have, but still they develop cerebral small vessel disease. This is a summary of the term of our result. We speculated maybe the missense mutations, if they form the trimers, if these mutation has located at the protein domain, it causes CARASIL. But the mutation located in the HCl activation domain, L3 or LD, or this mutation affect the trimer formation. This mutant has a possibility to cause a CSVD even in the heterozyte. But the problem is a nonsense or family system mutations. We still do not know the rules which type of the mutation causes symptomatic carriers or not. So the mutant which cause CARASIL, these kinds of mutant just has lost the protease activity only, but they activate the besides HL1. And therefore, in the heterozygote state, they still have the 50% of the residual protease activities. But the mutation which have been found in the symptomatic carriers, these mutants could not activate the beside HL1. Therefore, the net of the protease activity should be more lower than the 50%. And the mutant, if this it cannot form the trimer, the protease activity is very low. Therefore, we called CARASIL and symptomatic carriers with HL mutations. Maybe this type of the phenotype is related to the residual HL protease activities. If the residual protease activity is very low, it causes CARASIL. But still, less than 50% or near the 50% of the HL activities also act as a risk factor for the development of cerebral small disease. And therefore, we tested all of these kinds of diseases is HL1-related cerebral small vessel diseases. For the pathologies, the pathology is a loss of the smooth muscle cells in the CARASIL, and internal membrane is disparated in these CARASIL patients. But the most interesting thing is vessel is not occlusive. vessel is dilated. Therefore, in the small vessels of the calcium, the vessel is not is more dilated because the smooth muscle cells is degenerated. It causes dysfunction of the small vessels. This pathology is very resembled to the pathology of the sporadic type of the CSVD. This figure is obtained from the Professor Okeda. He looked at each small vessels from the cortex and the white matter, and he found even in the sporadic type CSVD, the neural cells is degenerative and the diameter is dilated and not occlusive. So from the pathological findings, the sporadic CSVD and the CARASIL resembles in terms of the intimate settings and also smooth cells and luminary dilatations. It's quite very different from the small vessel disease pathology, which caused by hypertensive cerebral hemorrhage. Also, in the manifesting carriers, symptomatic carriers, the neuropathology, small vessel pathology, looks like the same as calcium. So why these kind of dilated vessels cause diseases in these individuals? This is my hypothesis. Recently, the two unique functions has been identified. One is the elimination of the protein. The other is activity-dependent blood supply. Among them, the activity-dependent blood supply is very unique property for the small vessels. And about 84% of increased blood supply is regulated by small vessels. In small vessels, a pericyte or specific type of smooth muscle cells regulate the activity-dependent smooth muscle cell, activity-dependent blood supply in the brain. Therefore, we can speculate in calcium patients after the degeneration of these type of neural cells, activity-dependent of the blood supply may disturb in this individual. It causes some kind of dysfunction in the nervous system. This is a summary of the molecular pathogenesis of calcium. Calcium is caused by loss of external protease activity and then accumulation of the substrate of the HCl1 in intima. Many different types of the substrate has been identified, but among them, I would like to emphasize the two substrates. One is the TGF-beta, the other is the fibronectin. These two substrates is highly accumulated in the intima. After that, degeneration of neural cells happened, and then dysfunction of the neurovascular unit. And to treat these patients and also to treat the sporadic type of CSVD, one strategy is increasing the atrial protease activity is one strategy. The another strategy is decreasing the accumulated substrate like fibronectin or TGF-beta. Therefore, we are now screening the agent to achieve these kind of the functions. This is my collaborator. Thank you for your attention.